This video I am making you for just to make you familiarize with so many instruments you used in your research activities, but you should be knowing two things about them, that for what purpose you are using that particular instrument and what it is called as. So I'll try to just brief you about these instruments. If you have any query in detail, you want to know the mechanism how it works, then we will discuss this in upcoming, maybe in upcoming lectures. So this first one is heater and magnetic stirrer. This instrument is used for heating and simultaneously dissolving the substances. So it has both heater as well as stirrer. So some compounds needs to be, they dissolve at high temperature, or maybe temperature is required for their dissolution. So you can use this, and the inserted probe is there which will monitor the temperature. Next is magnetic stirrer bar retrievers. So this is long, you can see these are used to retrieve the stir these magnetic beads which you have inserted in your solution. So these beads are used for stirring, but if you want to remove that beads and make you want to further add some other another chemical into it and make the reaction complete, but you don't want that bead to be there because stirring part has already completed. So how you will remove that bead? Because maybe maybe you need not to insert your hand or directly into the solution, because the chemicals will also cause any reaction. So that's why these magnetic stirrer bar retrievers are used. This is conical flask. You all know, maybe this is used for titration purposes, where the volume is mentioned, and it provides the base of this conical flask is such that whatever reactions will happen, then base will provide the higher surface area. Next is micro tips. So these are used for pipettes. These are of different sizes, like 5 ml, 10 ml, as per the pipette size and what amount you want to retrieve. As per that, the 5 ml, 200 microliter, 1 ml, 10 microliter pipettes are available. And if you see this is a curved shaped micro tip. This you can use in loading your samples in gel for wait, for western blotting, because it needs the sample to fill in particular bill. So you can use there this type of uh, tips. Next is cage grill. This cage grill means this particular lid, you you can call it. So this cage grill are of two types. Like this upper one is used for closing purposes, and this cage grill is used in case of when you want to collect your, maybe if you want to uh, keep your animal there in cage, and you don't want that animal to, or don't have any, suppose you have kept feed here, and that animal might eat the feed remaining in the cage there, or maybe any contamination. In order to avoid that, these cage grills can be used. These are mainly used in animal houses. Next is blotting paper. The blotting paper you used in whenever there is sacrifice of your animals, and you want the this tissue, whenever you remove any organs and wash it with saline. After that, you want to weigh that organ, but uh, the organ should be sufficient dry, that it should not have that saline into it, so it will add more volume into it. So in order to dry properly, you require blotting papers. Next is Hamilton syringe, that is used for, this is Hamilton syringe, and it is used for stereotaxy apparatus. Whenever you want to do stereotaxy surgery, this surgery is used when you want to inject your drug or chemical compound into the directly into the brain of particular animal. So this syringe is used there. Next is gloves. These are also of different types, like white gloves that these are latex gloves, these are nitrile gloves, these are vinyl gloves, and of different types. But suppose latex gloves has powder inside it, or there these gloves may be reactive to some of the people depending on their sensitivity. So these gloves are preferred when you want to isolate RNA DNA. In their that cases the sterile gloves will be preferred. Next is aluminum foil. So aluminum foil has different purposes or different usage in our labs. Like whenever you try something in hot air oven, then you can uh, use this as a cap, or if you want to mm, prevent your drug substances to from chemical or lights. So light sensitive chemicals can be stored in aluminum wrapped uh, particular containers. So aluminum foil can be used for multiple purposes in labs. Next is polypropylene mice cage. So this is white cage. So this is made up of polypropylene. That's why they are called this as polypropylene cages. So this can be used for mice, rats, depending on the availability. Then disposable masks, as due to corona pandemic, we have seen that mask has many usage. Means in various conditions we can use, but mainly because we want to protect ourselves from fumes, gases, particles, viruses, any bacteriological things, or so many things. Next is paraffin wax. So paraffin wax is of two types. What comes in market is this is pellet form, and then some paraffin will come in directly compact form. So I'll say that whenever you want to prepare these histopathological blocks, you just go for this non-caking paraffin wax, because it will be easy to whatever amount you require, that you can take, and then only that much of amount of this paraffin wax you can melt. Otherwise what happens is, if you have whole bottle of this, then what you do is generally you keep a full bottle of this paraffin into hot air oven, and it causes you want that paraffin to melt, and that you keep in this particular temperatures, and after repeated storage and retrieval, it causes the means deformation of this original properties of this paraffin wax. So I would say that please prefer this non-caking paraffin wax. Next is disposable caps. So in certain cases you want your, in order to maintain hygiene, basically you don't want your hairs dandruff to fall into, suppose for example if you are analyzing DNA, so you know that even minute amount of hair particle will also affect your results. So such cases you want to have disposable caps. Next is disposable shoe cover. So whenever, if you are working in animal house, so you should wear actually this mm, shoe cover, because it will prevent uh, first of all anything to come into contact with your shoes, and the same you can, after use you can throw this into your disposable dustbin, and then just you can go with your, or if you are in hurry and you don't want to use your laboratory shoes, then you can just use this disposable shoe cover onto your shoes, and just can enter for, if you have to make entries several times a day in this particular area. So in order to avoid this again repeatedly tying, and this you can use disposable shoe covers.